quick disclosure, I finally have a startup company, and last year, fall, we call Nanogenics. So we have to disclose this. So for those of you who have not been introduced the concept of Porphysome Nanotechnology, so I will give a very quick introduction. Porphysomes are self-assembled from a single porphyrin-lipid building block. You can see the porphyrin ring conjugate to a lysophospholipid, and this porphyrin-lipid can self-assemble in more than 80,000 units to form these bilayers of the porphyrin. The consequence of that is when you have so much porphyrin absorb so much light, everything is quenched. Now your only way to dissipating the energy is by the heat. So porphysome naturally is a very good photothermal and photoacoustic imaging agent. When the porphysome dissociate, and they will restore their porphyrin monomers, the building blocks function, which is the fluorescence-guided surgery and for photodynamic therapy. For photodynamic therapy, porphysome is as good in terms of efficacy as a photofrin with FDA-approved drug. For the fluorescence-guided surgery,
the slide's not working.
Yeah, now it's working. So fluorescence-guided surgery, because they are in the cancer cells, they start porphyrin structure got disrupted, and they becomes a very big, good, smart reporter probe to guiding the surgery. When the porphyrin ring can be chelating metal, that means you can chelating trace amount of radioisotope, allow you to do the PET imaging, showing this left-hand movie, or you can label all the porphyrin ring with a paramagnetic metal, allowing you to overcome the sensitivity of MRI. Recently, we also show the copper-67, another radioisotope, which is isosteric to the copper-64, can be labeled, and they can form the very nice for SPECT imaging, and more importantly, is the beta particle can using for radiotherapy. We also recently demonstrate the porphysome can also be activated not just by the light, by the X-ray, and that leading to the anti-tumor efficacy we refer to as radiodynamic therapy. And another interesting development we observe is a porphysome photodynamic therapy in the quite astonishing result in the photoimmune therapy. We can induce many interesting immune response. So that will help the porphysome eventually go beyond local therapy. So in essence, the porphysome is, as in a nutshell, is a single porphyrin-lipid molecule self-assembled into liposomal structure, allow you to all these function, so it's intrinsically theranostic. For the last 10 years, we also worked with building a whole family of the next generation porphysome, varying different size, different shape, different function, different composition, and you can really do so much with it. It's really expanding the theranostic. So I'm going to explain using a few examples in this lecture and talking about next generation porphysome as well. So go to the topic of today is we talk about porphysome nanotechnology beyond lab, beyond light, and beyond cancer. So the first beyond lab is really about journey to the first in human, which is a very long journey and a very bumpy road, but we are really getting close to the finishing line. And that is not possible without all the funding support, especially from the Canadian funding agency, from the Terry Fox, from the OICR, from many of these, including NMIN. So a few interesting, unique translation challenge. I'll give examples. First, porphyrin-lipid is a radioisomers. And in 10 years back, we actually observed interesting finding. That is, the porphyrin-lipid, the SN1, SN2 isomers, they have a complete different radioselective biodegradation pattern. In the serine, they are exactly remain stable. But in the spleen and the liver, SN2 isomer having gone complete biodegradation. And that lead to an interesting radioselective biodegradation. We got a nice paper, but that leading to ultimate headache for the Health Canada and FDA, because either they recommend is either you're creating a pure isomers, or you're actually making the mixed ratio of isomer from your scale up for different steps, from the laboratory all the way to scale up, and which is impossible to do. So eventually it take a couple of years to figure out the scale up synthesis, a single porphyrin residual isomer, which is SN2 isomer. The reason we select that is because this one can go entirely biodegradation after the taking up the body eventually. So it's truly biodegradable. The second challenge we're facing is we're using interesting for the light therapy. So skin photosensitivity becomes an interesting phenomenon that we have to address. So for this one, we actually did the entire solo simulation to really characterize, quantify the photosensitivity of using Porphysome in all different scenarios. And we observed Porphysome has much limited skin sensitivity because of the low skin accumulation, and actually only happen in the deeper layer of dermis. So intrinsic multifunctionality of Porphysome allow us really a chance to design this novel umbrella approach called phase 1A, 1B for the inhuman design. So in essence, we can allow us to do a very low dose PET imaging for the phase 1A in advanced ovarian cancer patient. Once we have that, then we can actually showing the Porphysome safe in patient, then we actually can expand to all different sites and starting with the first in human theranostic studies. So the rationale for phase 1A, 1B is unique to Porphysome because corpus 64 can fit within the porphyrin ring of the Porphysome in a very stable form. And because it's only need to be labeled such a small amount, it's behave as a true radio tracers. Being radio tracers, the corpus 64 Porphysome and the Porphysome, the parental Porphysome, they are physicochemically, pharmacologically, toxicologically equivalent, as demonstrated in this slide. And that allows us to using the corpus 64 Porphysome to serve as a dosimetry tool to design, to inform the starting dose for phase 1B. For example, last year, my student Alex, together with the Bo Wan's group, was showing we actually developed a deep learning model. It's called NanoMask, which actually validate in the preclinical corpus 64 Porphysome PET data set, which can predict the pharmacokinetic output with a few PET-CT contouring. That is very useful. Actually, you only need a few PET-CT imaging image. You can allow you to predict the entire pharmacokinetic output, which could be very useful, especially in the patient. Another thing interesting is we also show corpus 64 Porphysome has a capture the Porphysome tumor selectivity and distribution quantitatively. So they're very good for dosimetry planning. So with that, we are now where we are in the phase 1A in collaboration with BC Cancer and especially the pharmacophore of the NMIN program in Marcel Bailey's leadership. We're able to actually get the GMP batch of Porphysome ready for phase 1A study. It has a 2-year expiration. We also have a 100 vial plan for the phase 1B study, and that is currently ongoing in the planning stage. For the radio label of the Porphysome, we are actually working with Dr. Ray Riley's lab at the University of Toronto in the pharmacy department, which literally is across the street. We are going to envision the Porphysome label with corpus 64 Porphysome there across the street. The 18th floor of the PMH will be administered to the patient. We have a GMP production of 1 patient dose of 200 microbecquerel every week. And this is a release table and the label. So we actually is ready for first in human. So beyond light, next. So copper is very amenable for labeling with a porphyrin. But porphyrin, if we want to label more metal, different metal into the porphyrin is limited. Because for example, if we want to label the lanthanide into the porphyrin, because the lanthanide ion is so big, it doesn't hold in the central cavity of the porphyrin. So people has trying to use different way to stabilize this porphyrin. But there's another different approach. That is the taxophrin, which invented by Professor Jonathan Sessler at Texas Austin, where he expanded the porphyrin from the 4-ring to the 5-ring that allow a 20% larger pocket and 5 coordination versus the 4 coordination. So this allows us to stable chelation of entire, almost all lanthanides into it. That is very useful because under the NMIN support, we actually develop a taxophrin lipid, which is self-assemble into a nanotaxophrin. Essentially, is a taxophrin version of the porphysome, allow you to stable chelation of 17 metals ions from the Simonian, European, terbian, and that actually be very powerful because allow us to, for the alpha emitters labeling, for the beta emitters, OJ electron emitters, and essentially entire nuclear massing space, we can starting to work on it. Just give you examples. We now work out a methodology to customize the metal labeling chelation at the different ratio, allow us to essentially control what metal and what ratio of metal to introduce. In here, we're showing the SPECT and imaging by the lutetium 177 taxophrin, India 111 taxophrin, which is good for OJ electron as well, and lutetium 175, which is cold lutetium, is good for PDT, and the gallium taxophrin is very good for MI contrast. More importantly, is actually useful for radiation therapy. We actually even develop a hot labeling, disposable kit for hot labeling using the 2 syringe pump. So this allows to disposable for all the radiolabeling procedure. This is done by the my former postdoc, Miffy, in the lab. So gallium and taxophrin being used for, as I mentioned, for radiosensitizers. They actually used for 1,000 patients in North America and European trial, but failed FDA approval because they didn't beat out the standard therapy. But implication for our point is gallium and taxophrin is very safe because otherwise you would not give to 1,000 patient test, but it's also very not a potent radiosensitizers. So my former student, Jiyun Shi, in the Peking University with Professor Fan Wen, thinking about how to moving this taxophrin, nanotaxophrin for radiation therapy to overcome by the one using the self-assembly. So you have 80,000 gallium taxophrin per nanoparticle to increasing the radiation therapy efficacy, or by carrying the oxygen. So for proof of concept, they want to put oxygen in the microglobin and loading the nanotaxophrin to synergize radiation therapy by relieving the hypoxia. And this method is actually becomes very useful. If you look at the hypoxia relief in both in vitro and in vivo, myoglobin, oxygen containing myoglobin inside the gallium and taxophrin really can relieve hypoxia in the tumor cells. As you can see, the most potent one oxygen relief is by this myoglobin gallium and taxophrin. Without gallium, they're still there. With myoglobin itself can relieve the oxygen. Hemoglobin is less efficient. But more interesting, in vivo, we really no longer need the myoglobin containing oxygen because the myoglobin gallium and taxophrin can pick up the oxygen directly from primary capillaries, and that allow to deliver the tumors. As you can see, hypoxia in the tumor can be relieved by this way. That directly leading to synergized radiation therapy response. As you can see the cholinergic acid, we can see the gallium and taxophrin already see the enhanced radiation therapy, but myoglobin containing oxygen, the oxygen containing gallium and taxophrin can really decrease the cell colonies, which demonstrate this is beyond the additive effect, it's a synergistic effect. In vivo, the gallium and taxophrin can also beat out the nitroimidazole, which is a clinically approved European radiosensitizer, and demonstrate long-term survivals. So another aspect of the beyond light is ultrasound. So on this aspect, my former student, Elizabeth, which came up this original idea of the porphyrin microbubble, essentially introducing the porphyrin-lipid into the microbubble. Microbubble are these gas-containing ultrasound responsive bubbles, and they only respond to ultrasound. So even with, if you have introducing the porphyrin-lipid, they allow you to multimodal microbubble, and they give you the fluorescence property, give you the photoacoustic property in addition to ultrasound characteristics. with this multifunctionality built into the microbubble that allow us to first observing the very first evidence of the micro to nano conversion. The porphyrin microbubble becomes nanoprogeny, become nanoparticles. And this is interesting because through this IV injection of the microbubble, right, infusion of the microbubble, then you irradiate the ultrasound at the tumor site, we observe very interesting phenomena. That is, the nanoparticle formed during this conversion process, they remain in the tumor for a long time, more than 2 hours. If you don't use this conversion ultrasound, then the porphyrin microbubble disappear instantly. 30 minutes later, they'll be all disappear. This is interesting because this means you can using the combination of the microbubble and ultrasound to producing this almost like an active EPI effect to promoting the delivery of the nanoparticles. That's the original indications. But we actually dive deep into this space and trying to figure out how this can be quantified. We quantify in a two method. One, we quantify in the shell fate of the microbubble. Because regardless the microbubble, or they become nanoprogeny, or they disrupted, the shell remain the same. And we have a perfect way to label the shell by using copper-64. So we label the copper-64 with a porphyrin microbubble that allow us to quantify what happened to this microbubble, the in vivo fate of the shell. So this, we demonstrate that. And more importantly, for the ultrasound property, we are really interested in what happened to beyond the vasculature, what happened. Are these nanoparticles with gas, and are they ultrasound sensitive? Can they still be using for ultrasound applications? For that, my student colleague, collaborating with David Gert's lab in the Sunnybrook, came up with this beautiful design, which is first design of you can allow you to simultaneously for the optical and acoustic monitoring of the conversion process by introducing a receiver underneath of the ring transducer, which will give you the ultrasound ring transducer, but now you have a receiver in the back, so you can basically monitor both the acoustic behavior and optical behavior. Using this methodology, she was able to determine the microbubble after conversion to the nanoparticles, nanobubble is actually contain the gas. So evidence of nanobubble was observed. That is the key evidence of that is at 35 minutes after microbubble dissolved, because 35 minutes there's nothing bubble left. The only thing remain whether they are the nanoparticle or not. So observe second harmonic generation, a very strong evidence of nanobubble. And that allow us to basically planning the new imaging and theranostic opportunity for this nanoparticle, acoustic response nanoparticle beyond the vasculature, for example, the photodynamic therapy process. So my last section of this one is talking about beyond cancer, because so far the Porphysome designed everything for beyond cancer, but the story is actually start from a cancer story. Then we actually accidentally came up with this antimicrobial PDT ideas. So one of the next generation porphysome, as I mentioned earlier, is so-called porphyrin-lipid nanoemulsion. As we design the porphyrin-lipids through the different self-assembly process, they give different giant porphyrin vesicle, porphyrin microbubble, or the lipoprotein-like porphyrin-lipid nanoparticles, and we are all text-free porphyrin. We also came up with the idea by introducing a nanoemulsion, the oil in water nanoemulsion, but self-assembled together by the porphyrin lipid. And this together is very similar to porphysome. The only difference is you still have pegolipid, you still have polylipid, you have the oil, which is glyceride triglycinate, as the oil containing. The reason oil is interesting is because it allows you to replacing that oil with lipidol, for example, which is a CT contrast agent. So far, the porphysome, we haven't done anything with CT contrast, but this can do that. And we can also introducing in this hydrophobic oil call, which carry in the oxalane, the antibiotics, all packed text of the drug. So this is a form of very nice core-shield structures. But what's most exciting feature is not this porphyrin-lipid nanoemulsion. Indeed, it is a big surprise finding. That is, in one of the control experiments we discovered, we don't need this porphyrin lipid at all. we don't even need a PEG lipid or anything. We can form this core-shell nanostructure with simply by two components. This triglyceride oil, which is widely used in cosmetic and food additive, and the porphyrin salt. And that can be coming from the off-shell porphyrin from many different you can buy from sigma-aldrich. Once we form this structure, we observe they have very high payload of porphyrin because essentially the whole oil is a porphyrin. You can carry different payload. But more impressively is we were surprised they are so collaterally, they're so stable, right? You can keep them for 8 weeks, 100-degree, pH 4 to 12, with a serine 50%, very harsh condition, mechanical agitation, nothing happened. They're very stable. We were actually very surprised by that. Then we actually dive into the mechanism. We realize compared to the polylipid nanoemulsion, the porphyrin salt nanoemulsion becomes very clear core-shell structure because of the porphyrin aggregation. As you compress by the surface tension, as you compress, you can see the pressure monolith actually from the compression, the oil going deep to the, going out and going deep, the partial phase separation. Once the partial separation happens, the porphyrin starting aggregate and then form very stable core-shell structure. This is the reason why collateral is very stable. But unfortunately, we were actually trying to using this emulsion for the cancer, but their in vivo, their clearance is very fast. So we were not able to get anything done because they're not suitable for IV administration. So we were thinking the place to use them probably is topical, right? And what's topical is used. Another important point is antimicrobial. So right before the pandemic, I visited São Paulo and I met my friends, Professor Bagnano and Karachi, and my former postdoc, Mbuza. And what we find out, we actually want to start a collaboration using this emulsion to do the antimicrobial PDT because they are the world expert in this field. So the first collaboration leading to a very effective result that we find out these called UPS, this is the porphyrin nanoemulsion, they are very, very potent in terms of PDT effect. 0.5 nanomole, a sub-nanomolar concentration, we achieve 6 log Q, which is very, very potent antimicrobial PDT agent. Then we got interesting, we studied more, then we realized it can lead into the bacterial biofilm can have a 6 log Q as well. And that actually is reason being that is we observe it is through this diffusion through this biofilm. Slowly, the porphyrin nanoemulsion will diffuse through this biofilm then to the Q. This is very contradictory to all the so far most of the PDT porphyrin agent for antimicrobial because they are mostly cationic, they are charged, so they can bind to the biofilm, then they act there. So this should tell us we may need actually a neutral porphyrin photosensitizer that allow you to diffuse through the biofilm, not trapped by the membrane that allow us to kill them better to take out the biofilm. So we move to in vivo in the wound healing model. We also observe the 6 log kill. So it's very effective in the multiple species and it can kill the biofilm, can kill the in vivo. So with that knowledge in hand, we design a brand new CIHR project. Is really our vision is want to do this hospital-associated pneumonia using the non-invasive deep lung immunization. Is a mouthful. But the idea is we want to, through the inhalation method to this lung with this pneumonia when the patient is under the intubation, so you can easily get in the inhalation through them. Now you want to get the target to the pathogen, you want through the navigating to the surfactant layer, and you want to deliver light externally to destroy the cancers, right? Destroy these bacteria. And you may ask, how can you imagine the light can deliver through the external body to this lung? And in fact, that is also on my question until I saw my collaborator and developed this method. They're using this so-called X-body light illumination device. Essentially, consists of hundreds of lasers in one panel. So it's a laser panel, not LED panel. And laser panel allow you to see this optical transmission through the rib cage of the pig. You can see this, the light can actually deliver the light into the lung homogeneously, but with different, you have to actually keep accumulation, because as the lung breathing, there is a distance of the light can go deeper or the more shallow. So this allows us to do, on average, can achieve 5 milliwatts per centimeter square light fluence in entire lung. And using the ICG, which has this wavelength of 800 AOA nanometers, allow us to actually treating phantom, which is putting the bacteria in the tube with ICG in a different location of the lung. Then you shine the light, you actually can lead into this kill. And this is so exciting. Actually, I was able to recruit the student who did this work to my lab as a postdoc, now carry out this work. Then we actually did the Monte Carlo simulation to test, is that wavelength dependent? We actually indeed, we observe by using the 400 laser from front or 200 laser each from the left or the right lateral or from front and everything, we actually observe the best with wavelengths to achieve this light transmission is exactly 800 OA nanometers. That is very exciting because now we want to have sensitizer for 800 OA nanometer that will allow us to do non-invasive light illumination for the first time to the human body, ideally. So ICG is the first one because it has a perfect wavelength, even though ICG is a very poor PDT agent. But unfortunately, when we try to make ICG nanoparticles, we were not emulsion, we were never able to get anything because it's purely quenched. Even you disrupt the nanoparticle, it's still quenched. We were actually surprised why. Then we realized is because ICG, they become a dimerized on the emulsion shell that actually subsequently form this J aggregate. We actually taking out this in vivo, taking out the emulsion, extract from the blood, we observe they actually become a dimer, right? Both the mass spec and MMR. They're really showing ICG is not a good option to do this emulsion because they become a dimer. They're not being a PDT agent. So we now are starting to, from the scratch, try to make a sensitizer with optimal wavelengths, 800 nanometers, high absorption, which is needed because we remember, even though we can get the light through the lung, they only have 5 milliwatts per centimeter square, very low, very dim. So we need to absorb as much light as possible. That means we want to use this agent has very high extinction coefficient. And you need a very high single loss gene, much more than ICG is less than 0.1%. And we're able to find this agent called silicon nephrocyanine. We're now able to making this compound has this beautiful matching wavelengths of AOE nanometers, has a very high extinction coefficient of 300,000, a 20% single loss gene, and that is very useful. So now we are working on all different aspects. We're addressing the light issue. Now we're working on the targeting using the peptide, using the nanobody, and we're working on the lung surfactant barrier because that's actually kills all the PDT agent. And we actually developed protocol to do this inhalation study, right? Using this using nebulizers. So I want to leave as much time for discussion as possible because this is what Professor Gilbert Worker was asked me for. So I will stop here and I will thank all my students, current and formal, particularly my first group of students. They're very well known as they invent a name called gangsters. And all my collaborators and the funding sources, and especially NMIN for the support. Thank you.
Gang. I have lots of, first off, it's a pretty amazing body of work. I've seen this now evolve over roughly 15 years or maybe 12 years. It shows how long you have to work on this to make it really go. It's not something that happens overnight or even in 5 years. So it's really terrific. I guess my first question, since you just talked about the lung and this photodynamic therapy in the lung, I was, of course, surprised that you get so much light into the lung, which is a good thing, I guess. Or I mean, not I guess, but definitely. But the other thing I was thinking about in pneumonia is in pneumonia, there's a lot of fluid in the lung and other kinds of sometimes things that contain a lot of polysaccharides and stuff like that. So how does the material get to the tissue that you most care about?
Yeah, that's a great question. And, I kind of allude to it. I don't have time to talk about it. One example is lung surfactants, right? We have demonstrated the PDT agent works for the antimicrobial, broad spectrally, kill gram-negative, gram-positive, kill everything. But as soon as you're adding, for example, a 5% Savanta, the lung surfactant, it will completely stop any PDT agent from working. So essentially, it got trapped in the surfactant. Means you actually, the surfactant is actually 70% lipid with the proteins. When you have a lipid nanoparticle, it's actually a disaster waiting to happen. That's why when you talk about inhalation therapy, even for gene therapy, beyond pneumonia, right? For cancer therapy or the gene therapy to do that, if you do a cystofibrosis, right, that is in the large airways. So they're very less, they're more mucosal, but less surfactant. That's like the surfactant layers. They will work. But when you are into the bronchial or these adenoid, these kind of areas, surfactant becomes a more prominent role. They actually become a barrier for all gene therapy. So we recently have a paper in Nanoletter really showing the surfactant barrier for all kind of therapy through inhalation role for the surfactant dominant disease, including pneumonia, including cancer. So we are working on several different ways to address it. And the one, for example, the alveoli-like exosome, we're talking about alveoli-like macrophage, and we're also working on the perfluorocarbon method. So there's many different ways actually addressing it. And so stay tuned. And this,
yeah,
this is not just a pneumonia problem. This is a whole gene therapy.
Right. The systemic, injection approach seems to be like something that you might connect to, the ultrasound or something like that. So that's really cool. The next question I have is, so you had this emulsion, which had, aggregates of the dye molecules on the outside, and you wanted to have this neutral particle so that it wouldn't stick so much, right? As I heard about that, I was thinking about another kind of emulsion. It's called which doesn't necessarily have charge. It's called a Pickering emulsion. A Pickering emulsion is one where you have a droplet of one material, and then it's almost like it's almost like a raspberry. It has lots of little particles on the outside, and that actually gives rise to the stability of the emulsion. And I'm just wondering if you guys have ever tried that kind of a structure, right? It's sort of it's like one step beyond your aggregate, right?
Yeah.
A little bit bigger.
It's certainly in my mind, but the one I really want to do is I want to using emulsion to carry oxygen, because one of the key problem of the antimicrobial of the biofilm is oxygen. Is a poor oxygen. It's similar for cancer, right? So hypoxia cancers, right? All the things we talk about. So emulsion give you a very good chance to carry oxygen. So this is what I will work next, actually currently working on, is making the emulsion with tons of oxygen, can carry oxygen in combination with, as I demonstrate, remember, with a taxophene, when you carry the microbe oxygen, it actually dramatically improves the radiation response. So if I carry the oxygen together with emulsion, then it addresses both the radiation response issue as well as the antimicrobial PDT issues. So that will be my priority. Pickering one is interesting.
It's just a different kind of emulsion, and it would also work for this kind of a idea that you want to have it neutral and so forth, right? I will encourage some students to think about some questions and to raise their hands when they are ready for that. Could you explain to me how this deep learning aspect of dosing and patient I guess I'm going to call it stratification is done, because I didn't get any really good idea from, you went so fast through it. I didn't understand.
Yeah, sorry, I was trying to control my talk within 30 minutes as
Yeah, sorry.
instructed. So the deep learning one. So first of all, anything about deep learning is you have to based on the data, right? If you don't have enough data set, then AI is not going to do much, right? So this stuff happened is when we actually had this achieve a 300 data set when we developed this microbubble radio copper 64 labeling to do all the mice study. We did a PET, we correlate with everything we do, the contouring, manual contouring. Then we thought, let's collaborate with Bowen to see if we can using this AI leading to the basic machine learning to help the very simply just do a contouring. Once we do the contouring, then we realize this is such a interesting aspect that is coloring of the image is nothing new. But if the imaging can predict the pharmacokinetics output, that's new. That's interesting.
Yeah, right.
So imagine we only did a few organs because not the beauty of all the data set is not complete, right? Some you do the liver, spleen, some is like liver, spleen, most of them we have it, but not all the organ we have them. But once we observe such a good matching or such a good prediction value of this one, then tell you we only need a few PET-CT scan, only a few organs that can predict the entire distribution of this one. That's first step. Second step, we can predict the PK that is out of that. So that becomes very valuable because no one need to sacrifice so much mice to do the biodistribution anymore if you can do a PET-CT. But obviously, the next question is, we work with microbubble. What happened to everything? Then we actually starting to work on with a data set of Porphysome. We work on the radio label antibody with the Stanford group. We worked with a multiple data set, different kind of construct, and we observe the correlation maintained. So then I become a big, I really starting to believe AI. Initially, like you think about it, right? You're talking about starting from a microbubble and you get exactly predicted value validated in the nanoparticle, you validate in the antibody data set, then all of a sudden it's become very generalized, right? So using is always want to see what the AI can bring you, something you cannot do before. Controlling is the first step. Predicting the PK is this idea for.
Right. So the prediction of the PK and so forth also, I mean, I think about cases where patients, may have to have multiple dosing, where the distribution can be different depending on exactly what you're targeting. And often, I don't know the toxicity level of this particular species, but I know many, things like doxorubicin and so forth, can be so toxic that a patient is really become sick sometimes from it. So this is a very powerful idea if you can carry it even further.
I can give you one example in the clinical case we plan to do. We are talking about copper 64 is 12-hour half-life, physical half-life, right? And by the time we label and give to the patient, we cannot give more than 3 doses, right? Probably 2 doses at maximum, because by then there is no more signals, right? Not enough signals. But imagine you have 1 or 2 doses of the PET-CT in a patient. If without this AI model, you were still not able to predict the PK.
Right.
And that means, this is just a simple example. I think that will be very valuable. I think Diana has a question.
Okay.
Thanks, Gang. That was a great presentation and so exciting. There's so many avenues of further investigation and near-term development coming out of it. So congratulations to you and your team. I have a question. It's a little self-serving and it's not so scientific, but we're just about to write the progress report for NMIN for the NCE. And could you share with us your assessment of the value added of NMIN? You talked about using PharmaCore, Michael Valek is the vice president of the student network right now, and he just won this pitch competition. And obviously, you've received NMIN funding for your project. So obviously, NMIN couldn't possibly fund all the avenues of research you covered today, but what do you think were the 3 most significant contributions that that NMIN made to you, and that were value added in terms of your ability to move all these ideas forward?
I mean, sure. Obviously the pharmacophore without the pharmacophore, we will never get access to the Marcel Bailey's BC cancers investigation drug program. Without that program, we would not be able to, even though the funding from the NMIN is minimum, but that connection, right, is really give us the access to our own GMP facility. So now we have a Porphysome GMP material made. That's number one. Number two, the NMIN gave us the access to the what is called nanocore.
Nanocore, right?
Nanocore, and which we did a lot of work with the cryo-TEM facility and everything. We actually have some of interesting work published in that aspect, right? So these are the infrastructure-wise, which NMIN providing. That's one. Second, the NMIN funded a project, the text-frame project, right? It is give us the theranostic truly a radio theranostic space, right? This is what we ultimately want to do. Even though despite the radioisotope supplies, we always have this issue, the supply here and there. And, without this supply issue, we will be probably already in a much advanced stage in terms of how they changed it, because we demonstrate that nanoparticle theranostic, you can do the different radioisotopes, different radionucleotide, you can different applications, right? So that's one of the key thing we actually NMIN is supporting, the real supporting the research grants. And another one is the NMIN's training support is tremendous. I mean, you can see the Michael Valek is play active role in NMIN training, in the environment, and from the same idea, moving the porphysome from the simple phototherapy and the radioisotope-based imaging to now radiotheranostics, right? And that's the idea one being the pitch competition, right? And the reason is Canadian all of a sudden have the supply of copper-67. So, and we already developed porphysomes. So that means everything we develop copper-64 can be translated to copper-67, right? So we are now aiming really, starting to really facilitate to pushing a lot on the radiotheranostics field, which becomes a tremendous value in the international space.
Oh, fantastic. Thank you very much for sharing that.
Gang, may I ask a question related to Health Canada? Because you now have, of course, a design for clinical trials, which I guess means that you've been speaking to them for quite some time. And the question, I mean, we're beginning that conversation as well. And the question I'm having for you is really related to the complexity of your composition and the tolerance of Health Canada for that. Let's say you have, 3 lipids, and you want to introduce one that hasn't been, previously FDA approved, or you want to add a second new lipid. What's your feeling on how much of a barrier that increases to, getting them convinced?
So the simple question, Health Canada FDA is always very supportive of the new technology. So issue is not them. Because they will never deny a good technology, right? If you demonstrate enough, prove safe, then they will always support you. The bottom line is come down to money, right? If you have sufficient resource, for example, if you have companies who sponsor you, then you don't have any problem, because all you need is just to go through all the procedure of the GMP and the GLP toxicity, GMP production, and you have done enough of your check mark of every checklist, you will go through the trials because somebody going to sponsor your trial. The biggest hurdle for us is when we want to do it academically, naively, we want to pursue academically, then we have to rely on the grant source and everything. We basically moving one step, then we actually apply funding for next step. That's going to take a long, long time, right? So I warn people, try not to, to be too naive. Pushing the translation without the commercial sponsors is not a good way to do it.
So if you had to estimate, I mean, maybe you don't want to say this, but if you had to estimate just for one of the therapeutics that you have in mind, though I don't choose one that you care about, that you care about all of them, choose them as an example. To get it through the phase 2 trial at this stage, what do you think is a investment from idea to the phase 2 trial? Millions of dollars.
Minimum 12 million.
Minimum 12. So that's a lot of money. And how much of that do you think you were able to, I'm just sorry you don't have to answer the question, but how much did you raise from government and how much from company partner?
Zero from company partners.
Nothing from company partner.
Yeah, so everything we were actually moving.
So you told me that we should raise from company partner, but actually you didn't use the company partner. Okay. So you mean it could have been faster if you had had a company partner? Yeah.
Way faster. You can shorten by at least half.
I see. Yeah. Well, it's an amazing thing that you managed to raise such a large amount of money from government. So congratulations. That's a skill all of its own that's quite important. And in addition to the science.